Hi everyone. So today's lecture is going to be made in several parts. The first part I would like to talk about is PCR primer design. First of all, because it sort of correlates with one of the questions I asked in one of the quizzes, and because we just covered PCR as an important tool in molecular biology, and also because it's an important skill to develop if you are actually interested in pursuing a career in molecular biology, whether it's medical or scientific. So if you guys can see here, this is basically the idea of PCR primer design. You have to create two primers to amplify a certain product using PCR. There will always be a forward primer and a reverse primer if you're just setting up standard PCR. And because I've mentioned that PCR can be used for lots of different applications, the PCR primer design will then depend. There will be certain alterations in PCR primer design to match the application. You can add additional things to your primer depending on what you're trying to do, or you can position your primer in particular locations depending, again, what you want to do. If you're trying to do short nucleotide repeats, or short tandem repeats for finding the criminal on the crime case from the blood on the crime case, then of course you will be designing your primer against a particular short tandem repeat area of the DNA. What I would like to talk about today is how we can design a primer in order to amplify a particular gene. In this case, we're going to talk about oxytocin because, well, it's short and it fits nicely onto a slide. So oxytocin, just briefly, it's frequently talked about as the hormone for love, trust, lactation, and all kinds of other wonderful things. It's not actually the one hormone for both human beings and little mice called voles to experience love. That's actually how oxytocin was found. Voles produce it, and so do we, but we don't do experiments on humans. So dopamine and serotonin also play important roles. But oxytocin is a short peptide. So the gene for the oxytocin is actually encoded in our DNA. And if we want to work with the oxytocin hormone, or specifically if we would like to make it in the lab, we first need to have the gene for oxytocin. So how would we get that? We would get it by PCR. And just briefly stop, this is the actual peptide that is the oxytocin hormone. And you guys can see that there's only 1, 2, 3, 4, 5, 6, 7, 8, and 9 amino acids in this polypeptide, as well. And there's one sulfur bridge between the two cysteine amino acids that forms this as a cyclical structure. But that's not important for what we're about to do. So here's the sequence that I got from NCBI for pre-propeptide of oxytocin. So I already covered how to get sequences from NCBI, but let's do this again. So you go to the NCBI. So let's back up here. You go to the NCBI database. If you just type NCBI into Google, you will get there. Then you select the database you would like to use. There's other ways to get the sequence for oxytocin, but the easiest way is to do it. One way that I think is easier for you guys to see is to using the gene database. So here I selected the gene database, and now I'm going to type in oxytocin. And because I want the sequence for human oxytocin, not the one from the pig, or the rhesus macaque, or the field vole, I'm going to type in human. And I'm going to click search. Okay, so here's what the database search engine found for us. Here's the gene for the oxytocin receptor. We don't care about it right now. And here's the gene for the pre-propeptide for oxytocin. Because oxytocin, like many other hormones, undergoes a lot of trimming and changes and modifications after it's been transcribed into mRNA and translated into protein. So here we have the gene card, the gene file for the oxytocin from human, and if you scroll all the way down past all the correlations, you'll find the NCBI reference sequences, which is actually what you can just search the reference database, but then it's a little bit more tricky to get this particular gene. So here NM stands for the mRNA, so I'll just get the mRNA of the gene. The reason I'm not getting the cDNA of the actual genomic area of the oxytocin gene is because it contains introns and it's really large. It's when we try to express something, scientists never work with the DNA version of it because it contains introns. We usually try to go the mRNA route, convert it to cDNA, and then you go from there. It's a process that hopefully I'll get a chance to talk about later in the course when we talk about transcription. So if you scroll all the way down, actually, never mind. So here you got the gene bank file for the mRNA of oxytocin. And it's a pretty long file. At the very end, you will have the peptide, the protein sequence for it, and a nucleotide sequence for it. But the other way to get the sequence to click on FASTA. Here, this little link. And it'll just give you the FASTA sequence for this gene. The word FASTA just means that there is this command line in front of the actual nucleic acid sequence. Don't worry about the command line. We'll just copy and paste this nucleic acid sequence into my PowerPoint presentation, which I already did right here. So this is a nucleic acid sequence that I got out of FASTA. Here you can also see these numbers just mean which numbered nucleotides are where. So here we're at nucleotide 10, and here we're at nucleotide 190. So in order to design primers against the oxytocin pre-propeptide, next I get the other strand. So we started with the coding DNA strand. That's what we got. That's what the FASTA sequence from NCBI had. And then I used my own software, but there's online tools that you can use to get the antisense DNA strand. You actually don't need the whole gene antisense strand, but it will help you guys right now to visualize how I design primers, or how you should design primers as well. So, how do we design PCR primers? Remember that we need a forward primer and a reverse primer for every standard PCR. The forward primer picking is easy. It's just if we're trying to get the whole gene, we're just gonna pick the first 18 nucleotides or 21 nucleotides of the gene. It's not the complementing sequence to the coding strand. It's the actual coding strand. So this sequence is the actual forward primer. There's no other changes that I'm gonna do to it. This sequence, the sequence on the antisense strand on the bottom, is the sequence for the reverse primer. So forward primer is 21 nucleotides long, and then the reverse primer, we just picked for now, is 21 nucleotides long as well. And you guys can see that I have skipped this large chunk of the gene here. Part of the reason I skipped it because you can see a long strand of A's right there. That's the poly A tail of the mRNA. We will talk about why there's a poly A tail when we talk about transcription. But if we're trying to specifically amplify the oxytocin gene from a library, we don't want this non-specific poly A tail. And then I skipped this area too because there's just a whole lot of T's in here. And this will make the melting temperature of the primer really low. So I sacrifice this chunk, and I'm just going to start right here with my trial PCR primer. So in this case, I picked them to be both the same length. So let's see what, sorry. And then, of course, another important part here is that right now, I just copy pasted this CAC-TTTTT-ATTTTT sequence right here. So the orientation here is 3', to 5 prime, because this is the 5 prime end, 5 prime to 3 prime, because the DNA is anti-parallel. You really need to remember this really well for the rest of the molecular biology course. So in order to get the actual correct primer as I'm submitting it to the synthesis site, I need to rotate it, right? Because the way you write DNA always is 5 prime to 3 prime. So here we go. Now the sequence has been rotated. Mind you, I did not complement anything. I didn't pick the other strand. I didn't change any single, no nucleotide was changed there. I just reoriented it. So to explain this even more, let's see what I did. Okay, this is how you reverse a primer. First, you copy the primer in the orientation that you happen to have it in. In this case, it's a 3 prime to 5 prime orientation because it's on the antisense strand and DNA is antiparallel. So forth. And then all I'm going to do is I will first write the correct orientation I'm going for. And then I'm going to copy the nucleotides one by one in the correct orientation. So first I will write the first nucleotide in the 5 prime orientation, in this case, is a T. So I'll write a T. Then there's an A, so I'll write an A. Then there's another T. And so that you guys don't get confused, because in stretches of T's, 4s or 5s, sometimes people miss a nucleotide. It may be helpful to cross them out as you copy them. There we go. Here's now the primer has been written out in reverse orientation, with T starting the strand because it's the most 5 prime, and the CAC finishing the strand because it's most 3 prime. Nothing in the molecule has changed. I didn't change anything about the information here. I just changed the orientation. Okay, so the next important thing about designing primers is you should not just pick the right region. In this case, I picked the very beginning of the gene and towards the very end of the gene. But you also need to make sure that the other rules of PCR are accounted for. So the typical rule for PCR primer design is you want a primer that's between 18 and 30 nucleotides long, give and take. Now, if you're designing something other than standards, typical PCR, if you have cloning applications and so forth, the sizes of your primers will change, and that's okay. The reason you want at least 18 base pairs is because you need to make sure that your primer binds exactly once in the genome, and 18 and up is the size that will assure that in the human genome it binds exactly once. If you're working with an organism with a far bigger genome, you will want to account for that, making longer primers. You also want to make sure that the melting temperature, and we will calculate it in a second, of the primers are within 55 and 75 degrees Celsius, and especially important that the primer temperatures are at least within 5 degrees of each other. And you also try to make sure that your GC content of your primer is within 40 and 60. So these are the rules for the most efficient PCR reaction. Sometimes we don't care if the PCR reaction is efficient, we just need to get that chunk of DNA. So right now, as I go through the rules, through the primer design, you guys will see that I will take certain liberties with the process, simply because the end of the oxytocin gene is so crappy. So you can, in this case, to calculate primer melting temperatures, I like to use the IDT primer analyzer tool. Different primer design tools available online will have different rules. I have already spoken about the NAB primer analyzer tool. In this case, it's a slightly different one. Serial Cloner also has a primer analyzer tool. And sometimes they will come up with different melting temperature for the same identical primer. And that's just, at that point, you just have a preference. I prefer IDT primer over, say, serial cloner. When I work with the enzymes from DNA polymerases from NAB, then I use their design tool. So hopefully this kind of helps with the understanding the thought process that goes into primer design. It's a bit flexible for certain applications. So I used a primer design tool here. I pasted these guys into this tool, and then I got a GC content for this primer is 67%. So there's 67% of the primer is G's and C's. That's what this means. While for this guy, you can see how many T's this primer contains. So the GC content for this primer is really low. It's 29%. It's on the lower side. Also, because there's so many T's and T's don't contribute much to the melting temperature of a primer. The melting temperature of the reverse primer is also very low. It's 45. It's way below the 55 and up regulation that we have. The melting temperature of this primer is pretty good. 63 sounds great. And obviously 63 and 45 are far apart from each other. They're nowhere near within 5 degrees of each other. Now, so the rule number 5 here says that we have to avoid runs of force of each individual nucleotide or dinucleotide repeat. And that's absolutely correct if you need a very efficient PCR product. But because we're working here with the very end of a complex gene and we want the whole gene, we will have to accept the fact that we will have runs of T's here. Because if we move any further in, we will lose a lot more of the protein, of the nucleotide sequence we actually want to amplify. But overall, because the melting temperatures are so far apart, this is not a good primer pair. So what do we do? How are we now stuck? Can we not amplify oxytocin? No. We will just adjust the primers to get the right melting temperature and hopefully the right GC content. So we will optimize them right now. So let's talk about first how we can potentially optimize the forward PCR primer. The GC content is kind of high there. We could try for a little bit lower melting temperature. And this is what I did here. You guys can see that the original primer has these 2 cytosines in the end, and I just removed the 2 cytosines of the sequence. This made the primer shorter. It's now 19 nucleotides. The GC content obviously went down. It's now 63% instead of 67%. And the melting temperature also went down. This is how much these 2 cytosines contributed to the overall primer dimerization, primer binding to the template. So I could go further. I could drop this A as well, but that would not contribute significantly to my drop in TM. It will have 0 effect on the GC content. In fact, it will increase it because now there's overall more G's because I took one A away. So going down to that A does not make sense to me. And I'm sort of getting into dangerous territory here because if you have primers that are 18 nucleotides and lower, you will have primers binding in several places in the genome, which I would like to avoid. I mean, statistically, there's a chance that the primer will now bind twice. Okay, so now I have this primer. What about my reverse primer? That was far worse. It had a very low melting temperature and very low GC content, simply because the ends of proteins, the ends of genes tend to have a lot of T's and A's in there because it's already kind of thinking about making a poly-A tail. It's a behavior I've found a lot of times in a lot of C-terminal regions. 3' regions of a gene. So what about this? What about me increasing the length of the primer? So I've took a whole lot more, like about 12 more nucleotides. And what I was going for here is, you can see that the previous primer stops here, and just adding that extra T will not help me. And hopefully these G's, however, because they have 3 hydrogen bonds to bind to C's instead of 2 hydrogen bonds of A's and T's, will significantly increase the melting temperature, and also this will improve the GC content that we're trying to achieve, which is between 40 and 60. So the GC content has significantly improved. It's now 41%, so that's within 40 and 60 barely. And the melting temperature now kind of matches the melting temperature of my forward primer. It's within 1 degree, which is great. This is what I really was going for, that my melting temperatures are really close together. And then, of course, because I added so much more to the protein, the length of the protein has increased, and it's above the 30 nucleotides recommended. But I will take my chances on the length in order to get my melting temperature just so. So here we go. Now let's look at our new 2 primers and the rules. So we're in rule number 1, the 18 to 30 nucleotides, we're sort of breaking it right here, but the forward primer is good. The melting temperatures are within 55 to 75, and they're within 5 degrees of each other. In fact, if I wanted to further play with this reverse primer, I could chop down some of the G's that would drop my GC content, that would make the primer shorter, and if I'm within 5 degrees, it would probably be okay for the PCR. And again, as I mentioned before, this primer definitely has 4 or more base pair runs, which is not good. But in this case, that's how the sequence of the gene looks like, and it's unavoidable in this case. And there's ways to help make sure that these particular runs of 4s are not a problem by adding additional things to your buffer, PCR buffer, which is a level of PCR knowledge that I don't expect you guys to have, and I'm not going to try to impart on you. But there's always more with PCR to get it to work just so. So anyway, we have a better primer pair. So are we stuck right now? Do we just believe it and go with it? No, there's ways to test the primer pair. I already talked about how melting temperatures are near and dear to my heart and that they need to be close. So these are the PCR primers we have. We can if you guys just look at the sequence and think about it just a little bit, you can totally tell me how long the expected PCR fragment will be. When we run this PCR reaction and then run it out on a gel, how big of a band are we looking for? So, any ideas? Well, it will be about 500 base pairs, simply because this thing says 500, and our reverse primer begins right here. So the only